So far, what we've talked about is what happens when a water molecule is split and we take the electron harvested from that water molecule and excite that electron using light energy or electromagnetic radiation. A byproduct is going to be that we make oxygen as well as ATP and NADPH. So what I've talked about so far is something that is often referred to as linear electron flow or non-cyclic electron flow. But before we move on, it's important to define also the role of cyclic electron flow. So again, what we've talked about so far would be considered non-cyclic electron flow. This is linear flow of electrons. It goes from the water molecule through photosystem 2 to the QB molecule to cytochrome complex to PC, ultimately to photosystem 1, ferredoxin, NADP reductase, and the electron ends up residing with NADPH. In so doing, we also proceed to make a bunch of ATP. Again, those electrons are originally coming from water, and we're exciting those electrons utilizing the energy taken from light. So it's important to keep in mind what exactly are we making here. We're making oxygen, we're making NADPH, and we are also making a lot of ATP. And this process produces similar amounts of ATP as well as NADPH. So you should be able at this point to map the flow of the electrons, which is really being able to describe what's happening by following the red arrow. Where do those electrons end up, and what is produced as a result of the flow of those electrons in non-cyclic or linear electron flow. So my thought question for you is that the Calvin cycle, this is where we're going to actually be making various types of carbohydrates and sugars. That process actually needs more ATP than NADPH. We need, for every 3 ATP that's needed, we only need 2 NADPH. However, non-cyclic electron flow or linear electron flow produces similar amounts of ATP. So what would be the result of only utilizing non-cyclic electron flow? Ultimately, we would end up with an accumulation of NADPH. And if you remember the role of NADH in the cell, it's a high energy electron carrier, and because it has carrying those high energy electrons, it actually can cause oxidative damage within a cell. So in general, we do not want to overaccumulate things like NADPH in the cell because it's going to cause damage to the plant cell. So what mechanism is in place for plant cells to avoid the overaccumulation of NADPH? Well, they have something called cyclic electron flow as well. So here, what we are doing is, not we, but a plant is doing, is they are creating only ATP utilizing this process. Additionally, they save water because they're recycling electrons when more ATP is required. So originally we would have gotten the initial electron from water. In photosystem 2, we would have excited that electron. It would have been passed on to QB and cytochrome complex, PC. Once that depleted electron reaches photosystem 1, the electron would have been reactivated and reaching that higher energy state with the infusion of electromagnetic radiation at P700. And at this point, normally P700 passes off the electron to ferredoxin. Now it's still going to pass it off to ferredoxin, but normally, if you recall, ferredoxin would then transfer it onto NADP plus reductase. Instead here, what we are doing is the ferredoxin is going to transfer it back to QB. And if you recall, the electron now has high energy and can do more work, which is to pump more protons against the concentration gradient, and then that electron can just continue to get recycled, all the while doing a bit of work, which is increasing the H+ concentration here in the thylakoid lumen. Now, if you remember, what was the role of creating that proton gradient within the thylakoid lumen? It was really to drive the synthesis of ATP, as that proton force was, the protons were flooding through ATP synthase and driving that proton motive force such that large amounts of ATP would be made. So in effect, when you do cyclic electron flow, it results in the synthesis of only ATP, whereas when we did the non-cyclic electron flow, going from photosystem 2 to QB, cytochrome complex PC, photosystem 1, ferredoxin, and then the NADP plus reductase to form the NADPH, that would result in the formation of NADPH as well as ATP. So again, this process, cyclic electron flow, does not create NADPH. Instead of shuttling that electron to NADP+, we're going to just shuttle it back to the cytochrome proteins. And in effect, we are still going to be synthesizing large amounts of ATP. And again, the reason we're going to do this is because more ATP is required in the plant cell than NADPH. So please take a minute and answer in your notes the following question: why is using cyclic photophosphorylation an advantage to a plant cell over using only linear or non-cyclic electron flow? So pause the video here, take a second, and answer that on your own. All right, so hopefully you came up with something along the following lines. So it's important to remember that with non-cyclic electron flow, electrons are going to begin at that photosystem 2, and they're eventually going to transfer to photosystem 1, and then ultimately reside in being transferred to NADPH. And this is considered a linear process. And again, non-cyclic electron flow is going to produce both ATP and NADPH. Cyclic photophosphorylation, on the other hand, or cyclic electron flow, is when we're going to be cycling that electron back through the process. And this is going to end up in the production of a high proton gradient inside of the thylakoid lumen. And so we will only be producing ATP. Photosystem 1 electrons are going to get excited, they're going to release that energy, and eventually that's going to return to photosystem 1.